Today, we will be presenting on our study muscle fiber type switching via TRPM2-mediated calcium signaling. In this study, the researchers manipulated a specific calcium permeable cation channel, TRPM2, to promote muscle fiber type switching in mice through exercise. Skeletal muscles have a remarkable capacity to undergo adaptive changes in response to use and disuse, including changes in fiber size, fiber type, and muscle force. Endurance training generally promotes slow twitch muscle types, while strength training promotes fast twitch muscle types. The two fiber types being studied here are type 1 oxidative slow twitch and type 2 glycolytic fast twitch. The presence of type 2 is evidenced by lactate in the body, which was measured in this study to find how much type 2 was present. We have two different sets of mice being studied in this test. Those with TRPM2 channels removed, titled TRPM2 knockout, and the wild type, which was left with the TRPM2 channels intact. The aim of this study was to examine whether transient receptor potential metastatic 2, which is TRPM2, whether or not it plays a role in muscle fiber type transition during exercise. The goal was to decide, 1, whether skeletal muscle fiber type transition and mitochondrial function were different following exercise training of the mice, and 2, if a difference is present, what is the mechanism by which TRPM2 mediates calcium signals in skeletal muscle during exercise? A key factor in the mechanism of fiber type switching is NFAT-C1, the cytoplasmic nuclear factor of activated T cells. NFAT-C1 is active when translocated to the nucleus by calcineurin, a calcium-dependent phosphatase. It is an inhibitor of fast-twitch muscle fiber gene promoters. So this means when NFAT-C1 is activated by calcium, it inhibits the switching of slow to fast-twitch muscle. Why TRPM2? TRPM2 is a calcium permeable cation channel that is found in many places in the body: the brain, the pancreas, the heart, the immune cells, and skeletal muscle. It's involved in a lot of different processes like cell proliferation, insulin release, cell motility, and cell death, but its role and function in skeletal muscle isn't that well understood. But given that it is a calcium permeable cation channel, the researchers speculated that the exercise abilities of TRPM2 knockout mice would be impaired. Their hypothesis as to why would be 1, a change in muscle fiber type, and 2, downregulation of mitochondrial function.
The experimental design that was used was that they took 12-week-old mice, and there were two types, ones that were the wild type and the TRPM2 knockout group. And these were then split into two separate groups of non-trained and trained mice. The training that the mice went through consisted of 60 minutes, 5 days a week, running on a treadmill at 12 meters per minute. And they did this for 12 weeks in total. And during this, the mice were then put through an exhaustion test to test how long they could run on the treadmill. And those times were recorded. But the exhaustion test consisted of a set speed at first at 6 meters per minute, and then would increase 1 meter per minute every 6 minutes until the mice reached exhaustion. And exhaustion was defined in the experiment as the mouse falling off more than 10 times in 1 minute. After the final exhaustion test, though, the mice were then taken and dissected, and their muscles and everything were put through many tests and experiments. Some of these were to find the mitochondrial potentials, the ATP levels through the muscles, and many of these were done through electrical stimulus in the muscles, so that way they could see the action potentials in action going through, and they would do calcium analysis of the muscles on the mice. They also did many other tests on different parts of the organs just to make sure that there weren't any inconsistencies, but there weren't other inconsistencies in the mice, and it was all taking place in the muscle.
Looking at our results, which are actually pretty exciting, we're looking at our first graphs here, which are the exhaustion tests that were run for the mice before and after training. Looking at our key down at the bottom, blue is going to indicate our non-trained wild type. Red's going to indicate our non-trained TRPM2 knockout. Greens are trained wild type, and purples are trained TRPM2 knockout. The first graph, graph A, looks at the running time of the groups of mice. Looking at the non-trained wild type first, we can see that they're able to run for around 60 minutes. After training, the green, the trained wild type, increased to almost over 100 minutes, which is pretty impressive for the 5 weeks of training for the mice. Whereas if we look at the knockout mice, we can see that before training and after training, there's almost no difference whatsoever, which means that their endurance didn't increase whatsoever. Looking at graph B, we're looking at the running distance of the different groups of mice, which we can see similar results. The non-trained wild type, upon being trained, increased from 800 meters to almost 1600 meters, meaning they almost doubled their running distance. Whereas with the non-trained knockout and the trained knockout, we can see again, there's almost no difference, meaning that these mice, there's got to be something different about them. Either it's the knockout, the TRPM2 gene that we knocked out, or it's something different. The scientists wanted to take out all the things that it could be other than the TRPM2 knockout. So one of the main things they did is they weighed the mass of the mice, they weighed the skeletal muscle mass of the mice, and they weighed the heart mass of the mice. And what they were looking for is they're trying to see, are the wild type gaining muscle mass that the knockout aren't gaining, which would mean that the results of their test aren't very accurate because these wild type have larger muscles, which means that they have more endurance. What they found actually surprised them and went along with their results really well. They found that there was almost no difference in muscle mass, meaning that the wild type mice weren't stronger, didn't have more muscle, but instead that their fast twitch fibers were most likely switching to slow twitch, whereas our TRPM2 knockout were not switching. We can see again on this graph B, our non-trained wild type and non-trained knockout, we have really dark bands on the fast twitch fibers, meaning that we have lots of fast twitch muscle fibers and very few slow twitch fibers before training. Whereas after being trained, we can see that our wild type actually gained some darker bands around the slow twitch fibers, and our knockout didn't gain any more bands. Their bands stayed the same color, meaning that we don't have any more slow twitch fibers among the knockout mice. This leads us into our next graph, which is fast twitch percentage over tubulin and slow twitch percentage over tubulin. Looking first at our fast twitch, we can see our keys the same. Our blue non-trained wild type were around 100%, and then after training, we actually decreased, which is what we expected to see. Whereas our knockout, we can see we're at around 150% fast twitch, and then after training, we actually increased a little bit, which probably comes down to the measurement tools used and probably human error. But what we didn't want to see was a decrease. And that's exactly what we didn't see. We didn't see any decrease. There was an increase, meaning that we're not losing any fast twitch fibers upon being trained. Looking at slow twitch, though, we can see where the difference lies. Our non-trained wild type before training were around 100%, but after training, they increased almost to 100%, meaning they almost doubled the amount of slow twitch fibers they had. Whereas our knockout, we see just barely an increase, which probably comes down to human error and means that we're really not gaining slow twitch fibers among the knockout mice. We can see this in picture form here. If we look at the non-trained wild type and the trained wild type, non-trained on the left, trained on the right, the bottom shows the slow twitch fibers taken from some of the mice. We can see the non-trained, we have slightly underdeveloped slow twitch muscles, whereas after the endurance training, the 5 weeks, we can see the slow twitch fibers are greatly developed and much more dense, whereas our fast twitch fibers, if anything, get a little less developed. Whereas our knockout, we can see that there's almost no difference if we take the non-trained muscles and put them under a microscope versus the trained ones. If anything, we have a slight underdevelopment of slow twitch fibers in the trained knockout mice, meaning that we're not seeing any switching in these two knockout mice between the non-trained and the trained. Now, looking at our next thing, TRPM2 lets in calcium, which activates the translocation of NFAT-C1. So if TRPM2 is knocked out, then we really shouldn't see any translocation of NFAT-C1, which plays a part in muscle fiber switching. This graph is looking at that NFAT-C1 translocation and how much of it translocated, how much of it didn't. So looking at our key, blues are control wild type, reds are control knockout, green is our electrically stimulated wild type, and purple is our electrically stimulated knockout. Electrical stimulation just means we took the muscle, electrically stimulated it to stimulate action potential so they could see where these proteins and enzymes and different things were moving when we activate those muscles. We can see in the wild type, we have around 100% in the cytosol, and then the translocation, we had almost 200% translocate out once we electrically stimulated the muscle from the wild type. Whereas our knockout didn't have any translocation when we did our electrically stimulation and showed the control group. There's almost no difference between the two. So where is NFAT-C1 going? That's the big question. And this graph kind of shows us exactly what's happening with this NFAT-C1. So looking at our control group, almost all the NFAT-C1 is found in the cytosol, barely any in the nucleus. But upon electric stimulation, we can see that it moves in the wild type from the cytosol to the nucleus, whereas in the knockout, it doesn't, meaning that we're not getting the calcium needed inside the cell to translocate the NFAT-C1, which would mean that our TRPM2 plays a major role in this pathway of switching muscle fiber types. And that leads us into one of the most important things from this research study, and that is TRPM2, when it's knocked out, means NFAT-C1 is not translocating, which means we're not going from fast twitch to slow twitch fibers, which is what we wanted to see in the study. Looking at the calcium levels in skeletal muscle, we should see that the blue wild type is higher when it's electrically stimulated compared to the red knockout, which is exactly what you see. The blue graph on the left, the peaks for calcium levels are higher when it's electrically stimulated when compared to the red, the knockout on the right. This shows the same kind of results when we look at mitochondrial calcium levels. In the wild type, when they're electrically stimulated, the blue, we can see that it goes much higher, around 200, versus the red, the knockout, that when that's electrically stimulated, we're around 175, which means that in the wild type, not only are we getting more calcium in there, what that means that we're going to have more mitochondrial activity, which means we're going to have more switching from fast twitch to slow twitch and much more oxidative energy.
In conclusion, we know that NFAT-C1 resulted in muscle fibers switching from fast twitch to slow twitch fibers. We know that the knockout mice exhibited more fast twitch fibers during the 4-week treadmill training. One of the things that they tested in the knockout mice was the production of lactic acid in the muscles. We know that fast twitch muscles needs quick energy, and they get that from glycolysis. When you use glycolysis, you use it anaerobically. Anaerobic glycolysis produces pyruvate, which leads to the lactate as a product, which consequentially is lactic acid. That's produced, that'll be put in the blood vessels. Another thing that they found was muscle plasticity and how it can easily be shaped. And that is dependent on the calcium signaling system. We know that through the endurance training with repetitive muscle contraction, it leads to muscular plasticity in response to metabolic demands. This results in an increase in slow twitch muscle fibers. Another thing that they found was the ATP production in the mitochondria was decreased, and that is because it is dependent on mitochondrial membrane potential. That was also decreased, so it slowed the mitochondrial ATP synthesis. And that also helps us understand why it used glycolysis more, because that's where it was getting its ATP energy from. Fast twitch muscles also diminish in size a little bit quicker. So if you have a higher amounts of TRPM, if you have less amounts of TRPM2, then you're going to have more fast twitch muscles, and they could atrophy quicker. So we think it would be interesting if we were able to manipulate that and protect individuals that were predisposed to muscular atrophy to help protect them from that. Another thing that was mentioned a little bit before in this presentation was that they tested the weights of the head, or the body weights and the heart weight and there was no significant difference. There also wasn't a significant difference in activities in their cages before, during, and after the experiment.